guys, so we are going to try something new for this genetics lesson. I've been told that if I try to do a video in some of the classrooms, there's a really bad echo, so we're going to try to do this in my office and see how it goes. Honestly, today's lesson shouldn't be too crazy hard for you guys because everything we're doing today is something that you've actually done before freshman year. And if you didn't get it freshman year, you're definitely going to have another opportunity to get it in practice before you are tested or quizzed on it. So the first thing I want to kind of go over are the two packets that you should be getting today. Okay, so one of them looks like this. All right, just has Punnett squares at the top, and it's got a bunch of note information on the front. And as you flip through it, you should see that there are just going to be some practice problems all throughout. Okay, so this is something you're going to need to keep handy, and we're going to be using it all throughout our genetics unit. Okay. Now the other packet that you're going to be getting, and I'm going to encourage you not to work while you're supposed to be doing notes, okay? This right here, it has Dragon Genetics at the top. In fact, you did a lot of these same problems freshman year. But this right here is what's going to be kind of like your graded activity. This is going to be your independent practice that you're going to do on your own. And so the Dragon Genetics is going to be, at some point, it will be for a grade. But kind of like the notes, it's something that we're going to be working on throughout. So the way the unit's going to work is you're going to do notes, and then we'll do some genetics problems, and then you'll have an opportunity to practice those, and then we'll quiz along the way, and some of those genetics problems are going to show up on quizzes, and then you will also see genetics problems when we get to the actual test. Okay, it'll be like in multiple choice format, and you'll have an essay that we'll talk about when we get to what the essay covers. Okay, so today what we're going to focus on is the note packet. Okay, you can work on Dragon Genetics on your own time. And one of the biggest things with genetics is if you're struggling, you're going to have to get help on it. All right, whether that be watching videos, whether that be getting help from a friend, going to claim. If you are struggling with the genetics problems, you need to get help because they're only going to get a little more involved, and they are going to require you to have an understanding of genetic concepts. And so if you don't have that understanding, it's going to make these very difficult. So like the lesson that you did yesterday where you learned vocabulary like genotype and phenotype, genes and alleles, dominant, recessive, homozygous, heterozygous, all of those are going to be essential to being successful in doing Punnett squares. Okay, so a couple of things I want to go over, and I realize that the purple is not going to be a good color for this, but one thing that I wrote up here is that Punnett squares equals probability. So it's really important to understand that the purpose of Punnett squares is to look at probable outcomes, what is likely to happen. That does not mean that is what is going to actually happen in terms of offspring. This is just giving us an idea of what could happen and what are the likelihood that certain phenotypes will appear or genotypes will appear. Okay. Now, this whole front page of your notes is exactly that, okay? These are your notes. You can write on them. You can write whatever you want to. You can add. This is where you're going to be doing all of your practice. So if there's anything that's said today that's helpful to you, you might want to write it down. You might want to have some scratch paper out just to kind of work genetics problems. There's some room in the packet, but there's some, maybe you want to write more than what space you have available, okay? And so the front part of this, what I consider, this is me, this is not necessarily essential rules to Punnett squares, but just over the years of teaching it, to me, for a basic Punnett square problem, there are kind of 5 basic steps, okay? And these are the 5 basic steps that I wrote up here. We'll come back to them here in a minute, but I do want you to see that they're right there, okay? On your note packet, that's one of the first things at the very top of your notes are kind of the 5 basic steps to doing a Punnett square. Below that, you have some important vocabulary. All of that vocabulary was covered in your notes yesterday. Here's a little section about ratios, and we're going to look at those as we do some practice problems today. And then down at the very bottom, you have some notes about types of inheritance or different types of crosses. Okay, so today what we're focusing on is a monohybrid cross. And a monohybrid, mono means one, hybrid is referring to our genetic cross. And a monohybrid is always looking at one trait. Okay, so what are the chances of having freckles? What are the chances of having curly hair? What are the chances of the flowers being purple? Okay, we're just looking at one trait at a time. Now when it comes to your basic monohybrids, you can do that for any type of inheritance. For today, we're going to be looking at your basic dominant recessive, which is your Mendelian genetics. That's going to show us those Mendelian ratios, just that basic simple, one allele is dominant, one allele is recessive. We're also going to take a look at incomplete dominance as well as codominance. And both of those are non-Mendelian. They do not fit those ratios that Mendel saw with his pea plants. Okay, and so we're going to see how they're different. We're going to work them a little bit different on each one. The main thing is we're going to have a third phenotype that we don't have in a basic dominant recessive. Okay, before we get started on actually doing the crosses, I just wanted to talk about the Punnett square itself. Okay, so when you're doing a Punnett square, all right, the outside of the box represents the parents, and technically it represents what alleles could be in the parent gametes. So remember, during meiosis, law of segregation, parents have two alleles for a particular gene, but they're only going to give their baby one. And so those alleles, those two alleles, are separated during meiosis, and so only one of those alleles is going to be in the gamete. So the parents could either give this allele or that allele, and that's what we're representing over the boxes, is what could they possibly give to the offspring. Now inside the boxes represent the possible offspring genotypes. Once again, possible, this is all based on probability. What could the babies get? So if mom gave this allele and dad gave that allele, this is what the baby could be. And so a lot of times it's just understanding the vocabulary and understanding what is the question asking me for. Alright, so let's go ahead and take a look at these steps, because I am going to rip this paper off and then we're going to move on and we're not going to see it anymore. You can always see it on your note paper. But number one, I think it's very important to always make a key. I know that that may seem very basic, and I know that a lot of you guys, especially if you understood them freshman year, you could probably even do some of these in your head. I've been doing Punnett squares for many years, and if I'm doing a new problem, I'm still going to make a key. I think it reduces, honestly, work in the long run. I think it also reduces errors and mistakes. So I would highly recommend that for every problem that you come across where a key is necessary, I would make a key to represent what is my dominant trait, what is my recessive trait, whatever it is, make a key if you're doing a Punnett square. The second thing we have to do is figure out what do the parents have, because we can't figure out what the kids are going to get until we know what the parents can give them. Okay, so we have to determine the parent genotypes. What letters do the parents have? And in a typical Punnett square problem, the question, or there's going to be kind of some background info, there's going to be some introductory info. They're going to have to tell you. They're going to have to give you the information needed to figure out what the parents have. Then we're going to separate our alleles. That's where we separate those letters. We take the genotypes and we split them above and beside the box. We actually perform the cross, and that's where we fill in the offspring in the middle. And then the last thing is to answer questions. Now this is one of the biggest mistakes students will make, is they don't answer the right question. So it's always really important to pay attention to what is the question asking me. All right? Let's see how this goes. All right. So what we're going to do is we are going to start in your practice packet. So we're on this paper right here. If you would, just flip it over. And we are going to start with the Punnett square practice. Okay. We are on the part that says monohybrid, dominant, recessive. Okay. So the very first type of cross that we're going to do is just a basic. This trait is dominant, this trait is recessive. This is what we call Mendelian inheritance, okay? So we take a look at those problems. We're going to do these together. You're going to have an opportunity to practice. And then hopefully you'll have some time in class to maybe go back and start working on your practice that's independent, okay? So we're going to start off with number one. And it says, in pea plants, tall is dominant to dwarf, okay? Now we should have learned in our lesson yesterday that if it's a basic dominant recessive relationship, we represent the dominant allele with a capital letter. The recessive allele is represented by a lowercase letter, okay? And so before we do anything, our first step is to make a key, right? So it tells us tall is dominant to dwarf. Now you can use any letter of the alphabet that you want. You could use the letter A for every trait that you do. I usually pick a letter that's somewhat related, but it really doesn't matter. The only thing I would suggest is try not to use letters that look the same, like the letter S, the letter C. If you're going to use a letter like, say, W, make sure that you can tell the difference between a capital W and a lowercase w, okay? So what I'm going to do is I'm going to use the letter T for tall, but you can use any letter that you want. So in our problem, it tells us that tall is dominant to dwarf. So if it's a basic dominant recessive, I'm going to use the same letter for both alleles, one capital, one lowercase. And we know that tall is dominant and dwarf is recessive, okay? So that right there is step one. And that literally took us like 30 seconds or less, okay? If you were doing this on your paper, it would take you even less time because I wouldn't be explaining it. This is important, okay? Please do not skip this step, okay? A lot of times it does make it harder because you have to constantly go back and reread your problem. So just take a few seconds, make that key, and then you have a quick reference, okay? So tall is dominant to dwarf. Now, step two, and this is actually what letter A, the first question, is asking us. Step two is we have to figure out what are our parent genotypes. So first off, a genotype are your letter combinations. What alleles? So do we have two big T's? Do we have a big T and a little t? Or do we have two little t's? So if we continue reading the problem, it tells us a heterozygous tall plant, okay? So for letter A, parent number one is heterozygous. And hopefully we know from yesterday's lesson that hetero equals different. So anytime you see heterozygous, that means that your alleles are different. Okay, so parent number one is going to be big T, little t. So a heterozygous tall plant is crossed with a dwarf plant. So our other parent is dwarf. Now, if we look at this, dwarf is recessive. The only way that we can get a recessive allele is, or a recessive trait, is if we have two recessive alleles, okay? It doesn't have to tell us that it's homozygous or heterozygous, because the only way that that plant can be dwarf is if it has two lowercase letters, okay? Remember, it only takes one capital, and that's the trait that we see. So, letter A, what are the parent genotypes? Big T, little t, heterozygous, and little t, little t. Now, step three in our Punnett squares is to separate the alleles. And so, for a monohybrid, it's really simple, okay? If my first parent is big T, little t, it doesn't matter which one goes on the top and which one goes on the side, but I'm going to put big T over one box and little t over the other, so the parent can give either this in their gamete or that in their gamete. And then this parent here, little t, little t. Alright, and we just did step three. We separated our alleles. Parents had two, they can only give their babies one. Now, inside, our next step is to cross, and this is where we look at our potential genotypes. Now, inside are genotypes, and so typically in a genotype, the capital letter comes first, okay? So what we do is we look above and beside. It's just kind of like you're reading a grid, and those are the two letters that are going to go in that box. So that would be big T, little t. Same thing right here. I'm going to get a big T, little t. This time I'm looking above and beside. So little t, same thing here, little t, little t. So this represents our possible offspring genotypes, okay? So we just completed step four, which is to do the cross. The final step is to answer the questions. So now we can look at the other questions that are with this number one. So B, what percent of the offspring would you expect to be tall? So if we look up here, okay, big T means that's going to be a tall plant. It's dominant, right? Big T is going to be a tall plant. And then these two babies are going to be dwarf. So in terms of percentage, it's going to be 50%, okay? Now, anytime you're doing a monohybrid, you're going to have 0, 25, 50, 75, 100, okay? Now, the next question, and this is something that might be a little more difficult. It's not that it's hard, it's just normally students don't remember this as well. But it says, what are the expected genotypic and phenotypic ratios for the offspring? And so when we're talking about ratios, this is where we're putting how many. We're using a number, and we're going to separate these genotypes with colons, okay? Now, I always do it the same way. I try to be consistent, and anytime you're consistent, you're less likely to make mistakes. So there's actually some notes on this on the front of your note paper. If you flip it over, there's a whole section on ratios. So if it's asking for a genotypic ratio, that means how many do you have of each genotype. And this is talking about the offspring. So without even looking at my problem over here, okay, if this is my trait, I always have 3 possible genotypes, okay? I could get 2 capital letters, and then I'm going to put a colon. I could get heterozygous, and then I'm going to put a colon. Or I could get homozygous recessive, okay? So if you know from math, the colon, anytime you're doing a ratio, it's the word to, T-O, okay? So what I need to figure out is how many of each of these did I get? My numbers go in front of the genotypes. So if I'm looking at my offspring, big T, there were 0, okay? And I know that you may say, well, why do we even put that there if there aren't any? Once again, I just try to be consistent. If I always do it the same way, then it's a lot easier to remember how to do it. So I had 0 big T. I look up here in my offspring, I had 2 big T, and I had 2 little t. So my genotypic ratio is 0 homozygous dominant, 2, that's what my colon says, 2 heterozygotes, 2 homozygous recessive. Okay, that's how you write a genotypic ratio. Now the other one says a phenotypic ratio, and if you remember, a phenotype is what you physically see. So once again, without even worrying about what the problem is, if this is my key, I have 2 possible phenotypes. My plants are either going to be tall, I'm going to put a colon, or they're going to be dwarf, okay? And once again, we're going to put our numbers in front of the phenotypes. So if I look up here, my Punnett square, 2 of my babies were tall, and 2 of my babies were dwarf. So my phenotypic ratio is 2 to 2. Once again, that question is pretty important, because if you're taking a test and the test asks for a phenotypic ratio and you answer this, it's wrong, okay, because this is a genotypic ratio. So it's really important that you understand what is the question asking you for, okay? So just make sure we know the difference between genotypic, the genotypes, and phenotypic, the phenotypes. All right, so that takes care of number 1, okay? If you look on your paper, you do have a number 2. So I want you to take a minute. I'm not going to give you a whole lot of time because this is a video and I don't want to just sit here and stare off into space. But I want everybody, have your paper in front of you, I want everybody to try out number 2 really quickly. So number 2 says, if 2 heterozygote plants were crossed and produced 12 offspring, how many would you expect to be dwarf? Okay, so we already have our key. We've already done step 1, and so you just need to pick up with step 2. All right, so I'm going to give you a few minutes, or really just a minute or so, to kind of take a look at that, and then we'll look at the answer together. Okay. If you have any questions, ask Miss Petty, ask a neighbor, work together. But let's make sure we understand this before we move on. So I'm not sure if everybody's had a chance to finish or not, but just for video purposes, we're going to go ahead and move on. And so step 2 is to figure out our parent genotypes, and it says if 2 heterozygous plants, all right? So hetero means different, and once I know what I'm doing, I can easily combine step 2 and step 3. I don't always have to write the genotypes down. So if the parent is heterozygous, I know that they're big T, little t, and I can just go ahead and separate those over the boxes. So I'm just kind of combining step 2 and step 3. All right, step 4 is to do my cross. So in this first box here, I get big T, and big T, little t, big T, little t, and little t, okay? Now, just to kind of make you think back, because this is an important concept that you need to understand, remember this is Mendelian genetics, all right? So the very first cross that Mendel did was a homozygous of opposite character. So that would be like crossing big T with big T, little t, okay? All of his F1s came out hybrid or heterozygotes, okay? And so they all showed the dominant trait. Now, The F1s were heterozygotes. He let them self-pollinate, and so it is your F1s that produced your F2s. So anytime you cross two heterozygotes, that's like the same as doing that F1 cross, F1 times F1, okay? So two hybrids or two heterozygotes. And if you remember, the ratio that he always got in the F2 generation was a 3-to-1. So this right here is going to show you where that came from, okay? And every time you cross two heterozygotes in a basic dominant recessive trait, you're going to get that 3-to-1, okay? So now let's take a look at the question. The question says, if the plant were to produce 12 offspring, how many would you expect to be dwarf? Okay, so this is a different kind of question than what we saw on the first one. So first off, we need to figure out, okay, let's look at our ratios, or let's look at our percentages. So of my babies, 3 of them are dominant, okay? 1 of them is recessive. So 1 of them is going to be dwarf. So what we're looking at here is 1/4, or 25%. And so if there are a total of 12 babies, you could just do a simple ratio like that, and you should come up with the number 3. We would expect 3 of those plant babies to be dwarf, okay? If you prefer another way to do that mathematically, as long as you come up with the right answer, that is totally fine, okay? When it comes to genetics, you don't necessarily have to show your work, because whether you did it right or not is kind of evident based on the answers. Now, if there are Punnett squares, you do need to fill out the Punnett squares. Okay, now like on a multiple choice test, you may not necessarily have to show your work. You're probably going to want to do it somewhere, but you don't necessarily have to show it. All right, so that covers the first problem type that we're going to do today. Okay, now we're going to take a look at incomplete dominance. So now we're moving into non-Mendelian inheritance. All right. So incomplete dominance, if you actually flip back over to your front page, okay, just as a refresher, incomplete dominance is when the dominant allele doesn't completely cover, okay? So we're no longer having capital letter covers over small letter. We have a different type of phenotype that is produced because now the dominant doesn't completely cover, okay? And so what happens is if you get a heterozygote, instead of showing the dominant trait, the heterozygote is going to show a blend of the 2 traits. So anytime you see incomplete dominance equals a blend, okay, of the 2 traits. Now, we're still going to follow the same basic steps, all right? So, number 1, we're going to make a key, okay? And so if we look at our problem, we're just moving on in our handout here. In incomplete dominance, now it tells you you can use a capital and lowercase. That is completely acceptable. In fact, I think most people, if you were to look up how to do incomplete dominance, they're probably going to use a capital lowercase letter. But and you don't have to do it like me, because it's not basic dominant recessive, because we have a different type of inheritance pattern, I like to use 2 different letters. And that way it tells me that it's something different, okay? So what I do is I use a capital letter for the more dominant one, and then I use a lowercase letter for the other one. Really doesn't matter how you do it though, as long as you know it's no longer dominant recessive anymore, okay? So it says in the problem, in some plants, red is incompletely dominant over white. So if I were to make a key for this, I would say capital R is red, and I would say a lowercase w is white. And that way I immediately know just from my key this is not a basic dominant recessive relationship, okay? Now, next step is to figure out what the parent genotypes are. Now remember, we have a 3rd phenotype, okay? I no longer have just red and white. So the way I would get red is if I had 2 red alleles, okay? The way I would get white is if I get 2 white alleles. But this time, if I get 1 of each, it's no longer going to be red, because this is not a dominant recessive relationship, and RW is going to give me a new phenotype. It's going to give me a blend, and if I were to blend red and white together, I'm going to get pink, okay? So now let's take a look at our problem. In some plants, red is incompletely dominant over white. We have our key. What are the genotypic and phenotypic ratios? If a red flower, okay, so right there I'm going to stop. A red flower, the only way to get red is if I have 2 R's, Okay, a red flower is crossed with a white flower. Okay, so the way that I get a white flower is two W's. I'm still going to separate my alleles just like I did before. So if I'm crossing a red with a white, all of my babies are going to come out RW. Alright, so we just completed step 4. Next step is to answer the question. Okay, so in this case it says, what are the genotypic and phenotypic ratios? Alright, so just like before, on number 3, my genotypic ratio, I'm going to do all of my possible genotypes. I could get 2 R's, I could get an RW, I could get a WW. And we're going to fill in the numbers in front. Okay? It really doesn't matter the order as long as you have the numbers with the right genotype. And so if I look in my boxes up there, I got 0 of those, I got 4 of those, and I got 0 of those. Okay? I would also ask for a phenotypic ratio, so remember that this time I have 3 phenotypes. I can be red, pink, or white. And this time my ratio is going to match up. Okay? I'm going to get 0 red flowers, I'm going to get 4 pink flowers, and I'm going to get 0 white flowers because all of my babies were RW. Okay? Alright, so that takes care of number 3. Now once again, you have a practice problem. What percent of the offspring would you expect to be pink if 2 pink flowers are crossed? Okay, so take a second, try to set that one up, and we'll take a look at the answer to that in just a moment. Alright, so we go back to the question, and it says, what percent of the offspring would you expect to be pink if 2 pink flowers were crossed? So our parents are pink, so the only way to be pink is if we get 1 red and 1 white allele. So my parents are RW. Both parents were pink, so RW and RW. Okay, so first box, 1 plant baby. Next box. Alright, and the question asks us, what percent of the offspring would you expect to be pink? Okay, there's a pink baby, there's a pink baby, and so our answer here is going to be 50%. Alright, I hope you guys are doing okay with this. We have one more problem that we're going to go over today. Alright, so the last monohybrid problem that we're going to look at today is codominance. Okay, and once again, this is not Mendelian inheritance. We're going to see ratios that are different than what Mendel would have gotten. Alright, now this one, there is one way to do it. Okay, so if you're moving forward in your notes, our next set of problems, numbers 5 and 6, are codominant problems. And it says since both alleles are dominant, remember the key to codominance is both are dominant, okay? In codominance, both alleles are dominant, so neither one is going to cover up the other. If we get both, we are going to see both, okay? So for codominance, you are supposed to use 2 different capital letters because neither one is recessive. So you are supposed to use 2 different letters, and they are both supposed to be capital. Alright, so we're going to pretty much work this out. It's a lot like incomplete dominance, and this is where you just have to have the knowledge. You have to know if it says incomplete dominance, or if it shows you that there's a blending, you have to know that it's incomplete. Or they're going to have to tell you it's codominant, they're both dominant, or they're going to have to give you the phenotype where you're physically seeing both. So there's going to have to be some kind of clue, or they're going to have to straight up tell you that this is the type of inheritance pattern, okay? So once again, we're going to follow our 5 steps. Okay, the first step is to make a key. So in our problem, it says, in some chickens, feather color is determined by codominant alleles. Black and white are both dominant. Okay, so for my key, I'm going to use a capital B and a capital W. B for black and W for white. Okay, now it also goes on to tell us that black and white are both dominant, and a chicken with both alleles is what we call speckled, which means both black and white. They have spots, they're speckled, okay? So the question is, what are the expected offspring genotypes if a black rooster is crossed with a speckled hen? Okay, so next step is to figure out our parent genotype. So we have a black rooster. The only way to be black here is if I have two capital B's. Because remember, if I get one of each, it's speckled. Okay, so the only way to get black is to have two black alleles. Now, the other chicken is a speckled hen. Okay, so speckled. That means black and white spotted. And so the only way to be speckled is if you have a B and a W. All right? So there's my black rooster and my speckled hen. I figure out what my possible offspring are. Once again, we need to pay attention to what the question is asking us. In this case, the question said, what are the expected offspring genotypes? It did not ask us for a ratio. You do not have to give any numbers. If you do, it's okay. They just need to be correct. But in this case, all they asked for were, what are the possible offspring genotypes? Remember, the genotypes are the letter combinations. So when I look at my offspring, we can either get a BB or a BW. All right? So always answer the question that is asked. We don't have to mention genotype. We don't have to give any numbers or percentages. It just simply said, what are the possible genotypes? Now, I know this is easy to miss because it got put on the back of the paper, and every year I forget to go back and correct it. So if you flip your paper over, there's a number 6 on the back, and you really don't have a lot of room to work. You don't have to do a Punnett square for it. You can. It's not required. But it's a different kind of question. This is one of those questions that is not going to follow your basic 1, 2, 3, 4, 5. Okay? In fact, it's asking you to kind of think backwards. So in this case, it's telling us what the babies are. Okay? On number 6, it says, what are the possible genotypes of two parent chickens if they have black, white, and speckled chicks? Okay, so they're telling us that in the babies, there are black chickens, there are white chickens, and there are speckled chickens. Okay, and this is where you may just have to do trial and error. All right, now we know that this isn't the possible parents, so this time they're asking for, what could my parents be? Because if I cross a black with a speckled, I get black and speckled. Okay, I don't have any white babies. And so what you could do is just do trial and error. Now, for time purposes, okay, the only way that you're going to get all three, if we kind of were to go back to our incomplete dominance, if you remember when we crossed 2 pink, we actually got 1 red, 2 pink, and 1 white. And so if we want to see all 3 genotypes, the only way that that can happen is if both parents are heterozygous. Okay, so the answer to number 6 is my parents were both BW. Okay, because you can see here, there would be a chicken that was black, there would be a chicken that was speckled, there's another speckled chicken, and then we also give a possibility of a white chicken. Those are the only parents that would give us all 3 possibilities. All right, so that's where we're going to stop today. Okay, in the future, we're going to go over dihybrid, and we're going to go over other problems that are at the back of your packet. You do have a quiz tomorrow, and that quiz is going to be over your basic genetics notes as well as monohybrid crosses. Okay, so I know you don't have a lot of class time left. Okay, I'm going to finish this video in a minute or so. So you're only going to have about 10 minutes in class, or probably less by the time you start the video, to work on your problems. Now, you rarely have homework, okay? And this isn't official homework. This is not going to be turned in, but here's what you need to do. Between today and the time you take that quiz, you need to do some practice problems, okay? Now, eventually you are turning this packet in for a grade. So what I need you to do, sometime today, in claim, before you take the quiz, tonight, whenever, okay, you have 8 problems, all right? A whopping 8 problems that you need to take a look at before you take the quiz, okay? On the quiz, there will be questions about the genetics notes, kind of the vocabulary, but there's also going to be some Punnett squares. There will be at least 1 basic dominant recessive, at least 1 incomplete dominance, and at least 1 codominance, okay? All of them will be monohybrid, and they will all be similar to like what we did today in our notes. So you have several examples to look at. You can always watch the video again. There's lots of resources online. You can watch Amoeba Sisters if you liked Amoeba Sisters. There's lots of videos for help out there. Or when in doubt, you can always email me. You can always go into Claim. You can always talk to Miss Petty. But if you are struggling and you don't know what you're doing, you need to get help. Well, how do you know if you know what you're doing? You need to do your practice, okay? So you have 8 problems. Please get these 8 problems done. If you do this whole packet in pieces, it's not overwhelming. If you try to do this whole packet all at one time, it's a lot of work, okay? So between now and quiz, get these 8 problems done. Miss Petty is actually going to write the answers on the board, so that way as you do them, you can check your answers and make sure that you're doing them correctly, all right? So if you have any questions, guys, let me or Miss Petty know. I hope you have a great day, and good luck on your Punnett squares.